Funding for this program is provided by Annenberg CPB to advance excellent teaching.
Zoology, forensics, ecology. These are just a few of the scientific fields united through the use of one common tool, the subfield of systematics known as phylogenetics. Scientists construct phylogenetic diagrams to map evolutionary relationships and better understand life's diversity.
One cannot understand biology except in an evolutionary context.
In the second half of the 20th century, scientific advances revolutionized phylogenetic techniques, uncovering a new approach to the study of evolution and revising our understanding of life on Earth. Patterns abound in nature. We seek to understand the world around us by studying such patterns. But like the flat meditation garden, we can also impose man-made patterns to try to find natural ones. Another example, phylogenetic diagrams that help us organize and classify the various forms of life on Earth. They reveal telling patterns of information about how organisms are related to each other and how they have evolved over time. Further, exciting advances in molecular biology have uncovered patterns at a different level. A's, C's, G's, and T's arranged to encode an organism's genetic makeup. In most cases, genetic data lend support to the diagrams designed with the morphological characteristics in mind. In other cases, not. But in every case, we use them to learn more about relationships and the past that has led to those relationships.
Classification traditionally has been about putting things together that look alike. In our refrigerators, we put the eggs in one place and we put the milk in another place, generally to organize it. And we might have skim milk and whole milk, but we put the milk together. So in our daily lives and in biological science, we put things together that seem to be alike.
This is kind of interesting.
Philip Gingrich of the University of Michigan has been asking, where did whales come from? How did this mammal evolve to live in the water? And how is it related to those that live on land? To develop possible answers to these questions, there are two kinds of diagrams Gingrich can construct. A cladogram maps relationships by comparing the number of character differences between organisms. But as a paleontologist, Gingrich considers the appearance of these differences over time. So he uses the more traditional diagram, the phylogenetic tree. It offers a different perspective on how this is related to this.
Phylogeny, it's a metaphor referring to the venation in a leaf and the idea that life started with a single stem and then has branched into all that you see in the venation in a leaf. So it's to give a sense of a tree-like or leaf-like structure of the history. Phylogenetic reconstruction is trying to put together a venation-like picture that shows how the animals living today and the animals that lived in the past are connected to each other. If there are ancestors, they would be on the same vein, and if they're what we call sister taxa, they would be at the terminal branches of the different veins.
Gingrich used the fossils of modern whales and their prehistoric ancestors to analyze physical changes in the cetacean order.
We start with observations. We start with what we can see. We break things down into their smallest characteristics and then analyze these by comparing them with each other to find the most parsimonious possible history we could use to explain the distribution of characteristics. It's important to understand that the word phylogeny is used in the traditional sense going back to the 19th century of a branching diagram starting from a stem with the branches separating up through time. Time is an axis of a phylogeny. This is the way that we represent the pattern of evolution graphically. It's like drawing the positions of the continents on a map or something. And we plot something about the morphology against time.
Gingrich relied on this graphic diagramming technique to try to figure out how an order of mammals had evolved to live in the ocean. In the 1960s, paleontologists like Gingrich believed they had figured out the evolutionary history of whales. Their conclusions, represented by a phylogenetic diagram, were based on the fossil record of the time. They proposed that the cetacean order, starting with the Archaeoceti, or archaic whales, evolved from a group called the Mesonychids. But because the fossil evidence was scanty, not all researchers bought this idea. Some thought archaic whales evolved from a different group, one that also gave rise to hooved land mammals like hippos.
For the last 20 years, I've been really consumed by the idea that we have to find all of the links, all of the transitional forms from whales living today back into the transition from land back into the land mammals. This idea about mesonychids being at the base of whales has been a good one, but we've long known that what would cement it would be finding the ankle bones of early whales. There are two parts of a mammal that are most informative about what it's related to. One are the teeth, and the second one are the ankle bones, because they're complicated. They preserve well as fossils. They're dense bones. They bear a lot of weight.
So, Gingrich searched the Valley of the Whales in Pakistan, hoping to find ankles that link cetaceans to mesonychids and show that they are indeed most closely related to each other.
If we could find those, we could corroborate, I thought, the idea that archaic whales evolved from mesonychids.
Molecular researchers disagreed. Those who give precedence to molecular data say even with the prized fossil, this picture of whale evolution would be inconclusive. They argue DNA evidence should be considered whenever possible in phylogenetic reconstruction. The very nature of DNA makes it a document of evolutionary history. It's a molecular record from parent to offspring. Comparisons of the DNA sequences of organisms provide a perspective on relationships unavailable if only fossils are studied. Since species evolve by the gradual accumulation of mutations, the amount of nucleotide sequence difference between a pair of different genomes should indicate how recently those two genomes, and therefore species, shared a common ancestor. To determine proper relationships, researchers compare the data of no less than 3 different organisms. They scan thousands of base pairs to identify points of divergence in the sequence. Base pairs that differ among all 3 organisms are excluded from the data set. Researchers then compare the remaining nucleotides to determine which 2 sequences are most similar. The 2 species with the most matches are most closely related. While Gingrich is convinced he can find the ankle that's shaped just right to link whales with the extinct mesonychids, molecular data work against him, supporting the opposing view of whale evolution.
Our molecular colleagues have been increasingly strident that whales and artiodactyls have to be closely related. And they even think that they can see where whales came out of the artiodactyls, and they propose that hippos are the group that's closest to the ancestry of whales. But I never fully believed it because molecular people can't see groups that are extinct. And it would be very easy for mesonychids to be in the middle and them not to be able to see them, just looking at living animals. So I started in Pakistan looking for these. And finally, in the year 2000, we found whales that the predators didn't steal the hands and feet and tails. And we found that the ankles of whales are not like mesonychids. And I can tell you that for the first week after finding this in the field, I was in denial because I couldn't believe that the ankle bones are exactly like artiodactyls. I couldn't believe it.
Gingrich's new fossil corroborated the very hypothesis he was trying to refute.
Mesonychids are out of the picture, and whales and artiodactyls connect to each other. Mesonychids are still there somewhere on the side, but they're not part of this story. In this case, now the fossil evidence supports, yes, a connection of whales and artiodactyls without any mesonychids at the base. And so our molecular colleagues are telling us many answers, none of which we can fully believe because we don't have evidence from fossils that tie them together too. So this is a very special case to be able to tie the groups together with fossils too. It's what we've been working to get for all these groups, and at least now we've got it for one.
Paleontologists finally agreed upon a phylogenetic diagram for the cetacean order when molecular data and morphology matched up. But when comparing microorganisms, it's often hard to find distinguishing physical characteristics. In such cases, including molecular data reveals the powerful significance of information contained within the genome. It can inform our view of all life on the planet.
Molecular data have made an absolutely profound revolution in the way the evolutionist can look at the world.
At the University of Illinois at Urbana-Champaign, Carl Woese used RNA molecules to transform our understanding of life's evolution and, by extension, all of biology.
Molecular sequences are an extremely delicate expression of the organism's phenotype. They are more subtle and more informative than any other characteristic the cell has. Without molecular data, most of the living world would not be understandable in taxonomic terms. When classical biologists defined the categories of organisms, they worked essentially with animals and plants. Now, animals and plants are actually only a small fraction of the diversity of life on this planet. And most of that diversity lies in the bacterial world. It isn't that microbiologists didn't want to understand the evolutionary relationships, the natural relationships among bacteria. It's that given the classical criteria, the shapes of the cell and the physiological reactions, they were unable to infer from these the relationships among organisms.
But Woese was interested in the evolution of translation and other defining moments in the origin of the cell, life's most basic unit. And this interest compelled him to ask, how are single-celled organisms different and similar on a molecular level?
When I began my work, the only relationships that were known were those inferred by the classical evolutionists, which were indeed the tree of the plants, the phylogeny of the animals, the tree of the animals. The whole microbial world was terra incognita. So the first thing to do is to create a tree that encompasses all of life. And it was clear at the time that this probably could be done by comparing the sequences of particular molecules. The one that was obviously the most useful, because it was the easiest to handle in the laboratory, was the one called ribosomal RNA. Ribosomal RNA is universal. It occurs in every living organism. And so began an extensive survey of the sequences of ribosomal RNAs. At that time, I'm talking here 1970-ish, one could not sequence the molecule outright, but one could cut it into pieces, separate the pieces, and then characterize each of the pieces, thus producing a catalog of fragments that were characteristic of the ribosomal RNA of a given organism. And this is the way we began. And we accumulated catalogs from a great many bacteria, say up to 400 or so. And these catalogs, when compared, would allow us to tell how close the various bacteria were to one another in ancestry. And from this, you could then build trees. through the usual tree-producing algorithms.
As the first researcher to compare bacteria at this level, Woese found one rather large group particularly mischaracterized in the five-kingdom tree of life, the archaea. For the first time, this group of organisms appeared so numerous and so fundamentally different from other groups that Woese restructured the tree of life to accommodate them.
By the time I finished, it was very, very clear that this was something of a different kind. It was neither of the so-called prokaryotic kind, nor of the eukaryotic kind. And it just didn't fit the signature of the bacteria or the signature of the eukaryotes. I realized, oh my God, we've made a mistake here in assuming that all prokaryotes were the same kind. Life actually comprised three major types of organisms, not two, not just eukaryotes and that's prokaryotes. But bacteria and archaea and eukaryotes, that was a really thrilling moment. Of course, right away then there was a rush to repeat the experiment on the assumption that something might have gone wrong somewhere. Well, it hadn't. They do now begin to define a signature of their own. That was thrilling. But it's thrilling to sit there and look at the universal tree for the first time. This is it. This is the complete picture. This was a major change in the way that you think, and you don't just say, oh, please think this other way. You say, the way you think is now stupid. You have to think this new way. You have to take into account phylogenetic relationships and do so in an all-embracing way. And people don't like to be shaken up like that. So of course, the new ones coming along just grow right into it and accept it. It's just the changing of the guard. I think Darwin was supposed to have said, the road to scientific progress passes through the graveyard.
A new picture of evolutionary relationships has broad implications well beyond the practice of systematics. Researchers can apply this comparative context to a wide range of scientific questions. The tree of life informs every subfield in biology, from genomics to ecology. Investigators construct phylogenetic diagrams to understand past and present life forms. Even in forensic investigations, tracking a correct evolutionary path can help identify microbial killers.
More than just a letter to Senator Daschle may have traces of anthrax.
At NBC News, a second employee contracted an anthrax skin infection.
In Florida, 7 more employees being treated for symptoms of anthrax.
The anthrax mailings of 2001 stunned Americans. It was immediately clear we had a long way to go in our understanding of deadly microorganisms. What most Americans didn't know is that scientists like Tim Reed of the Institute for Genomic Research had been studying the molecular foundation of Bacillus anthracis before the attacks began. A team of researchers completed the full sequence of the anthrax genome in 2002. They used that data to try to establish relationships among different strains of the bacterium. Reed hopes that a phylogeny can help trace the source of the strain that killed 5 people.
Molecular systematics is a major part of the war against bioterrorism. The idea is generally to give the evolutionary relationships between organisms, what the possible common ancestors were and what are most related.
Phylogenetic diagrams built according to changes identified at the molecular level tell the organism's evolutionary history. And when dealing with microorganisms, the evolutionary history is the best way to distinguish one sample from another.
Now that we have DNA sequences, we can really get to the root of evolution. The original people who built phylogenetic trees and studied evolution that way could only work with what they had available, which was essentially morphological differences in how the organism looked. It's much better now that we have DNA sequences to use those to investigate evolution. The project that we took was to sequence the genome of the anthrax strain used in the Florida attack and compare it to the genome that we were sequencing before that occurred. You can use genome sequencing to find markers like single nucleotide polymorphisms, which we call SNPs, and you can use those to construct essentially phylogenies or something close to it with a few number of SNPs to try and identify the source of the strain. You have to base evolutionary relationships on the DNA sequence. It's the most fundamental way of classifying organisms. Any other type of data essentially is a phenotype of some sort that relates back to the DNA sequence. When we had the Florida sequence, one of the hypotheses was that someone had inserted something into the Florida sequence to make it more virulent. So we did a search of all the sequences we found by genomic sequencing method to make sure that they matched the original genes. And basically we found no evidence that there were genes inserted into the Florida strain.
Unfortunately, finding no inserted genes gave Reed's team little to go on. Further complicating the search, Reed's phylogeny connects the Florida strain with one of the two main sources of anthrax. Samples from the source have been shared with labs worldwide since the 1980s, making it extremely difficult to eliminate possible suspects. But Reed knows that a comparative approach to molecular data still yields a variety of useful information.
We are going to sequence very closely related neighbors of Bacillus anthracis. And what I hope will happen is that some of the SNP differences we find in Bacillus anthracis will be shared with these near neighbors. And in that case, we can actually root the tree. We can see which of the Bacillus anthracis strains are more closely related to the ancestor. So which have evolved first and which are newest. Maybe we can trace the path of how anthrax evolved, where it came from, and how quickly it's been moving around.
Researchers study deadly microorganisms not only to trace their origin. Cures and preventions might also emerge from this work.
You can use the DNA sequence to find vaccine candidates. We can predict the type of molecule that might be on the surface of anthrax using the genome sequence. And the same sort of technique is available for designing new drugs. We can look at the genome sequence and we can say, this protein, I could design a drug and it will kill the cell if it hits this protein.
A phylogeny for anthrax may not answer every question we have about this particular bacterium, but it's clear that phylogenetic techniques can be used to great advantage in forensic investigations. Phylogenetic study uncovers the natural patterns of relationships among Earth's organisms, but revealing the universal tree of life remains a work in progress. Research continues in many areas. Carl Woese is interested in understanding the pattern at the base of the tree of life, a pattern laid down between 3 and 4 billion years ago. In this work, he must consider a specific type of gene transfer, something known as horizontal or lateral gene transfer.
Now, there was a time, apparently, before even one reaches the root of the universal tree, when the organisms were in very intimate contact with one another via horizontal gene transfer.
In lateral gene transfer, organisms pass their genetic information back and forth, not just vertically from parent to daughter cell.
The horizontal transfer of genes does not really bespeak the lineage of the organism. Genes came in at a certain point and made the lineage appear to change, but it was only the gene was bringing its own history with it, not the history of the organism. But then there came a time when at least some of the genes in the cell became essentially permanently fixed into the design fabric of the cell. And that time was the first period at which we can begin to represent what happened, this evolutionary course, in terms of tree representation. So at that point, you could begin to see permanent genealogies, and so that 3 billion years later, you could read them, as we do in the laboratory. And so that meant you could begin to represent the situation by a customary phylogenetic tree.
Today, we recognize the 3 domains, but some of the genes and traits found in eukarya have been passed on from bacteria and archaea, and are products of lateral gene transfer. And sometimes even more than small parts of DNA were transferred. Mitochondria and chloroplasts almost certainly started out as free-living archaea or bacteria that became intimately associated with ancestral eukaryotic cells. The division of the eukaryotes into the kingdoms we recognize today reflects the pattern of inheritance of these organelles. Although vertical gene transfer has become the dominant mechanism, lateral gene transfer still exists today, especially in archaea and bacteria. Indeed, it is one reason why antibiotic resistance can spread between different bacteria.
I think just about every reading adult is familiar with antibiotic resistance. And this occurs usually when a cell picks up a package of genes called a plasmid. Among those genes is one that produces a protein product that enables the organism to somehow avoid, inactivate, overcome the antibiotic.
Once science devised methods to identify and classify organisms according to permanent genealogies, the universal tree of life came together. And as the nature of scientific data itself continues to evolve, the need for an accurate and reliable framework for the study of organisms is as critical as ever. Phylogenetic reconstruction remains one of the best tools for the continued study of life's diversity and unity. It is one of the pillars of bioinformatics and has proven the best method available for comparing organisms, testing hypotheses, and integrating the disparate fields that comprise the study of all life on Earth.
Evolution is a unifying force in our understanding, and it does a good job of it.
If you have any idea how many organisms are living and how complicated they are, a person is easily overwhelmed. Without some way to organize that, to simplify it, to be able to talk about groups of animals in a meaningful way, it's overwhelming. And nothing can make sense without understanding the basic phylogeny.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.